a warm welcome to everybody who's joined us here in person today. Thank you for attending, and to extend a welcome to the speakers in our next session, which is titled Modeling Rare Disease with Patient-Derived Cells, a Platform for Discovery and Precision Therapies. We are pleased to have a panel of 4 speakers from leading foundations and biotech firms who will share their approaches and insights into this field. Our speakers will discuss how patient-derived cells can enhance the understanding of rare diseases and lead to more precision and effective therapies. So as we come out of the first session where we see the impact of these therapies, understanding how we can keep advancing research, accelerating the path to the therapy to get these important therapies to where they matter most. Just before we start, as a reminder, we'll take questions at the end. We have a Q&A session with the panel. For those online, if you'd just like to enter your questions in the chat, we will get to them during that session. So without further ado, I'll move to our first presenter, who is Dr. Irene Morgenstein. Irene is the Director of Preclinical Initiatives at the Children's Tumor Foundation, leading drug discovery in neurofibromatosis, or NF, as I'll refer to it from now on. Irene earned a doctorate in pharmaceutical sciences from the University of Sciences in Philadelphia and completed her postdoctoral training at the Rockefeller University in New York. Before CTF, she worked at generics, designing preclinical drugs for the central nervous system. She has managed data strategy at Regeneron Pharmaceuticals and as part of research and development IT team. Irene's expertise includes preclinical sciences, data innovation, and collaborations to advance treatments for rare disease. Her presentation is entitled Accelerating Drug Discovery in NF. Thank you, Irene.
Thank you very much, and thank you for having me here. What a wonderful event. First time attending and speaking, and I'm excited to learn from all the presenters today. So I'll spend the next 20 minutes or so speaking about our work at the Children's Tumor Foundation and how we put all of our effort into accelerating drug discovery for, it's actually neurofibromatosis and schwannomatosis. There are a few reclassifications recently, so hopefully I can clear those up in the next slide or two. For those that have not heard about NF, or collectively, this is neurofibromatosis and schwannomatosis. It's a family of progressive debilitating genetic disorders. 2 main types. There's neurofibromatosis type 1, and then there's a set of schwannomatosis. Within schwannomatosis, there's NF2-related schwannomatosis and non-NF2-related schwannomatosis. Collectively, tumors grow throughout nerves on the body, throughout the body, but that's just a very general way of speaking about it. For NF1 specifically, some of the manifestations could be disfigurement, blindness. Unfortunately, these benign tumors can turn into a malignant type of tumor called MPNST, malignant peripheral sheath tumor. You can have cognitive deficits and bone deformalities. And although it's a rare disease, it's actually not that rare. You can see by the numbers, 1 in 2,500 individuals. This is an unmet need. And I'm excited to say, as of last week, we actually have 2 approved treatments now. We had one in 2020 and another one last week. However, for schwannomatosis, there are no approved treatments currently. The incidence is lower, and one of the key manifestation in NF2-related schwannomatosis is hearing loss. So the tumor will grow around the 8th cranial nerve, leading to hearing loss and loss of balance, difficulty walking. And again, these are all progressive. Non-NF2 schwannomatosis, these patients have excruciating progressive pain. And again, there's no available treatments. Before I jump into, a few other slides, I wanted to show a video because it's always better to hear it from the patients and learn a little bit about their journey.
My name is Leanna, and I want you to see me and see an F.
My name is Blake, and I want you to see me and see an F.
My name is Deshaun. I want you to see me and see an F.
People who have NF are so varied in different types.
I basically tell them it's genetic disorder, can cause tumors to grow anywhere on or in the body, and it's like something that you're born with.
I look healthy on the outside, but on the inside I have over 100 tumors in my body.
But what people don't see with my diagnosis is that I'm losing my hearing.
They just see this person, oh, you're really in pain every day?
I have NF1. I'm the only person in my family that has NF.
They make assumptions that they're not just like every other teenager.
Because some people are different than other people.
I might see you as someone who's different, but actually understands you and knows a part of understanding of what you're going through.
All of my tumors are internal. I don't have any skin tumors.
One of the things people can't see about NF is the constant worry we have, knowing that a tumor could be growing anywhere in your body at any time.
I'm not too sure. I might develop tumors, I might not.
I think about the future and I get scared, but then I meet people within CTF and the NF community, and it's inspiring.
Just because I have NF2 and I've had 27 surgeries, despite these challenges, I'm still pursuing my dreams.
Someone could show a scientist, and then they could figure out a cure and keep on experimenting.
Although I have NF and I went through my own struggles, the second I found out my son had it, my entire world changed. And it was my mission from then forward to find a cure or an effective treatment.
The more attention there is to it, the more research, the greater chance they're going to find a cure.
We want everyone to support her and support everyone dealing with NF in the way we've felt so supported by the CTF community and the NF community.
My name is Jesse. I want you to see me and see NF.
My name is CJ. I want you to see me and see NF.
So what we do at the foundation is focus really heavily on drug discovery. So we have been talking about being the drug discovery engine for NF. And so what that means is we'll bring all the right people into kind of one space or, in collaboration. So we have access to patients, to clinicians, to researchers, to government officials. And how can we do this together? How can we bring those folks together to accelerate this process of finding new treatments for NF? And our patients are at the core of everything that we do. And our mission is simple yet complicated, and it is simply to end NF. And so that's a little bit about us. We are in the States, and we also have a headquarter in Europe. So we're in Europe and the US. So we've heard stories already kind of this morning and all throughout the session. I'm sure we'll hear more about how the patients endure more than a frightening disease, not knowing what will happen, when it will happen, how to treat it, how to handle, giving care to the children, the patients. But the system is also a little bit broken. And we won't get into every part of that system and what's broken and how for rare disease it's hard to recruit patients. You have multi-sites where you have to get patients from. You have complex protocols, but I will focus on preclinical. That's where I'm heading here. So what are some of the challenges with preclinical drug development? We know it's expensive. We know it is risky. We know that it often does not translate well from preclinical findings into the clinic. We know that some models, and I think I heard it mentioned, they're difficult to get your hands on. So even if you have a repurposed compound and you have a theory, what do you do with that? You need a model. How do you find those models? Sometimes it takes a while to negotiate those terms with institutions to use those models. That leads to very long timelines to initiate a study. Time is of the essence. So whatever we can do to shorten this and make it more cost efficient or effective, that's what we're trying to do here. And of course, standardization. If this is something we're going to use over and over again, these assays, we need to make sure they're standardized and they're used in a similar way. So in order to overcome these hurdles, something that we're working, and this is our newer initiative, it's called the NF-focused preclinical hub. And it's not a physical hub, it's a virtual collaboration kind of concept. And so it's a integrated network of experts, so various institutions, contract research organizations, and other tools. And the idea is to bring them all together into one central virtual space and make them accessible. So how do we make the models available, accessible for drug screening? When I say models, I'm talking about in vitro cell models, in vivo mouse models. We're talking about data tools. We even have pig models we've developed that can be used for PK/PD experiments. And drug libraries, right? How do we, look through, scan the literature? Because unlike other ultra rare diseases, there's a ton of literature around NF. So we've generated NF-focused drug libraries. And how do we source those compounds to make them available for drug screening? So this is our initiative. This is what we're working towards. But how do iPSCs, and that's kind of the focus next, how do they play a role in this? When we thought about which models to generate or build as part of the preclinical hub, we focused on clinical translation. iPSCs were one of the models. We have others. We're also working on PDX models we're trying to generate and put into the hub. But right now we're speaking about iPSCs. And I don't think I have to go into much detail, but we know that these cell types are patient specific, so we can kind of recapitulate the mutations that a patient could have. We can go ahead and differentiate them into disease-specific cell types to better understand the pathology in vitro. We can scale them, we can reproduce them. We can even use them for either medium or high throughput screening assays to screen some of the repurposed drugs that we've identified. And they have a translational potential. So here's kind of where, I think that's the button, yeah. So here's where we're fitting in here in this in vitro. We have other models that we're working on, as I mentioned here, but we're just gonna focus in on this little part. Before I talk about how we went about generating these cells, I have to give a little bit of background. So if we focus in, and this is on neurofibromatosis type 1, NF1, the gene is on chromosome 17. And this is a rather large gene, one of the largest in the human genome. It's 60 exons long. And then I think this number is actually outdated, the 2,800 different pathogenic variants. I think there are more reported at this point. And the protein product is neurofibromin, and it acts as a negative regulator of the RAS-MAP kinase pathway. So this is indeed a RASopathy. To have tumorigenesis in the NF1 patients, it's based on a two-hit theory. The first hit is a germline mutation in the NF1 gene here, followed by a somatic mutation in the second allele of the NF1 gene, and you have loss of heterozygosity. This can occur in different cell lineages, causing the complex spectrum of manifestations that I mentioned on the first slide. So it can happen on Schwann cells, astroglial melanocytes, mesenchymal progenitors, and these are just a few examples here. So you can quickly see how complicated this disease is. So how do we model this? How do we even start? What we did here with our first set of iPSCs is based on a publication that came out of University of Alabama at Brigham, UAB, we selected a germline variant based on genotype-phenotype study. The study identified 6 mutation hotspots, and we ended up focusing on this GRD region because this is important for RAS signaling. And not only did we focus on this GRD region, we also focused on a more severe phenotype. This more severe phenotype led to cognitive impairments, spinal neurofibromas. COMs are like pigmented spots on the skin and skeletal abnormalities. So that's how we went about, where do we start with these cells? And we worked with iXCells and their team here to generate our first set of cell lines that will go under this preclinical hub. We used CRISPR, so we did not start with patient generated. There are a few hurdles there we're working through. We started with CRISPR generated cell lines, and we used CRISPR to introduce a missense het knock-in with this arginine 1276 glycine variant, again identified from this publication. We then used the het cells to introduce a truncating mutation on the stop codon on the second allele. And I'm sure the iXCells team can tell you all about the wonderful work they did to generate these cell lines for us. So right now we're early on in the process. So I'm afraid I don't have too much data to share. It's just a concept that I'm sharing here. So right now, we are hopeful that these cells will be available through iXCells soon, as soon as they're characterized and validated. And we have big plans for these cell lines. We would differentiate them into, for example, Schwann cells progenitors. These are the main affected cell types in NF1. We would validate these cell lines and develop a high throughput screening platform using them and generate spheroids as well. We do want to go beyond this one specific set of cells. We have plans to look at other mutation types. There are frameshift mutations, splicing mutation, nonsense deletions, so many that we can generate. This is only the starting point. And we absolutely want to expand into schwannomatosis because for schwannomatosis, although the incidence is rather low, I think that's the area that really needs some of these new models. So we're starting here, we're moving forward to all of these other next steps. And for the drug screening approaches, we would go ahead and validate the initial using clinically relevant drugs. As I mentioned, for NF1, there are 2 right now, selumetinib and mirdametinib, that are on the market. So we would go ahead and start with those validation studies when we have the cells ready to go and generate, next gen 3D models and functional assays. These are our plans, long-term plans here. So, I'll put a quick pitch in for CTF in the preclinical hub, saying that, what we're looking for are vendors or biotech companies that want to work with us and be part of this preclinical hub, industry partners that are interested in NF or that need some of these resources. Again, we're developing these resources for the community, for the research community, for the pharma companies that need to use some of these models. That is our goal, is to make it easier to screen compounds for NF. Foundations with insights, I've already heard so many here that I jotted down, so always reach out. Happy to chat. And institutions that are eager to co-develop innovative models. I think that's about it. And I just want to thank you. We have a small but mighty team at the foundation. And here are most of them. And it really takes a village, and all of these individuals contribute quite, to these preclinical efforts and to the mission of the foundation overall. So just join us in Ending NF. I'm happy to answer questions later or talk about how we can work together and collaborate.
Thank you, Irene. As we heard there, the importance of gaining access to patient samples to improve our models is a key bottleneck and a challenge. And for our second speaker today, it's my pleasure to introduce Barbara Van Heer, who is president of patent.org, the Rare Cancer Foundation's online platform, which empowers patients to contribute tissues and data to research regardless of US treatment location. She currently leads all of the team's patient advocacy and institutional relationships, as well as manages patent.org's tissue collection. Barbara's presentation is titled From Consent to Collaboration, Practical Insights into Patient Tissue Procurement.
Thank you very much, and thank you, iXCells, for inviting me to be here today. And I am the president of Pattern.org, and Pattern.org is an initiative of the Rare Cancer Research Foundation. And the Rare Cancer Research Foundation was started in 2013 by a rare cancer patient who happened to be diagnosed with ocular melanoma, who was discouraged after his diagnosis to find that basically no treatments had changed in the previous 25 years before he got his diagnosis. And as he did some research and worked with other rare cancer groups, found out his situation was the norm, that very few of the rare cancer groups had had significant change in their diseases in terms of treatments for 20 to 25 years. So he created RCRF in order to build the infrastructure that could be used by any of those rare cancer groups and now also rare disease groups to help get rid of some of the roadblocks that might be stopping research from moving faster. And just for full disclosure, this rare cancer patient happened to be my nephew, Mark Labs. Pattern.org was created in late 2016. We did a lot of research with the different groups, the different rare cancer advocacy groups on what really were the challenges. We talked to a lot of researchers, what were their challenges? And the number one thing they came back and said is, we don't have access to patients and we don't have access to patient tissues. So we created Pattern to provide that infrastructure to be able to work with patients. Currently, it's across the US. It will probably be into Canada within the next year or so, but we are primarily US based now. So wherever they're being treated or wherever they're getting their care in the US, to be able to get tissues from them. For rare cancers, usually it's solid tumor tissue. For blood cancers, of course, it's blood, but we're able to collect virtually any type of patient tissue. And so how does Pattern.org work? Patients learn about research studies for their particular disease. They then go to Pattern. They are then matched to studies. In fact, Pattern is in the process of being updated and should be available, hopefully the new version within the next 6 weeks, where if there's more than one study that a patient can contribute to, they can actually rank their choices of how they would like their tissue to be utilized. It's not guaranteed that necessarily they'll get their first choice. Let's say if there's not enough tissue or there's something about their condition that doesn't allow them, but we will let them know where their tissue actually went. So they're matched to studies, and then they can go through Pattern.org online and consent to that study. If they have questions as they're going through that, they can reach out to us, or there's also a chat feature. Once they consent, we're notified literally within about a minute of that consent process. And depending on the disease that we're working with, whether we need to, if they're having a surgery, let's say it's a rare cancer, then we'll reach out to their clinical team at whatever hospital they're being treated at. It can be a large academic center, can be a regional hospital, it can be a community center. So we reach out to that patient's clinical care team and work through all of the challenges that many of you may know about trying to get tissues out of institutions to get them to release that tissue. The patient can talk to their clinician about wanting to participate in the study, not required. If they don't feel comfortable doing that, we're happy to take on that burden to get that tissue out for them. So then we coordinate the transfer of their tissue and data because we'll also get the clinical records associated with that patient's case. So some folks are confused, like what exactly does Pattern do and what do the researchers we work with do? So on the left side is Pattern. We handle the patient consent and the tissue donation. As I mentioned, we work with that clinical team to collect the tumor. And in case of rare cancers, we also get the germline tissue. If it's not a cancer, let's say it's a rare disease and we need whatever we need to collect for that, we will work through the details with the group that we're working with to determine what exactly we need to put in the collection kit and who we need to work through to get that. And then we handle the consent and also the clinical data associated with that particular patient. We have the full medical record, but we give de-identified information to the research groups that we're working with. Then over on the partner researcher side, they are the ones that develop the models and any genomic or other data. We get the tissue to them. If it's fresh living tissue from like a solid tumor, then we have it overnighted the day of the surgery. So it arrives by 10:30 the next morning. The only hiccups we have usually if there is a FedEx delay, like due to a snowstorm or something like that, but we will track it. We will let the research group know where it is along the way and when it arrives. And then the researchers we work with, we really, again, we were founded to help break down those roadblocks that were preventing progress. So we strongly encourage and in many cases require that the research groups we're working with share the data pre-publication. For biotechs, we make some exceptions, but we'll work with them on what data they might be willing to share. And then that data that is shared would go through public biorepositories. Also, any models that are generated, again, with biotechs, it may be slightly different. We require that those models go into like an ATCC or other biorepository. How do patients find out about us? We have a wonderful network of foundation partners. This is just a small list of the ones that we work with. We will work with actual foundations. We'll work with social media groups. And we're also in the process of working, setting up agreements with some smaller community hospitals where actually tissue from many of the procedures literally gets thrown away. And so we're working to try to set up a more effective system to utilize those tissues. Who are the research partners we're working with? The ones listed here are the ones that are currently on our website that patients can consent to, but there are 11 additional sites that we are currently in the process of onboarding. From the work that we've done, again, primarily it's been rare cancers, but we can help rare disease. 65 research models have been created and they are available and in the public domain or shortly to be in the public domain. We have collected from over 120, well, it's 120, I think the last count is 124 hospitals. And again, I should stress that the primary work that we have done to date has been with fresh living tissue, not with stored tissue, but we're also capable of collecting stored tissue. We've collected from 39 different states and 20% of our samples have come from underrepresented groups. So kind of an overview of what we're doing with Pattern and with RCRF. Patients contribute to research and empower themselves at the same time. We get those tissue donations. Historically, what we have done is we collect the tissue, whether it's a blood draw, whether it's tumor tissue, whether it's normal tissue, and then we send it directly to the research lab. We have recently been working with some groups that have asked if we could store tissue for them. Let's say they need 10 or 15 or 50 samples of a particular tumor or biological material. We now do have a biobank in conjunction with Precision for Medicine so that we can store the tissues there. We can also do different assays there. And so we'll work with the groups that want to work with us on, do they want the tissues direct or to go through the biobank? We collect the medical records. We're also encouraging, particularly for cancer patients, but for others that where it might be interested, is to get genomic sequencing. And so we're more than willing to help communicate to the doctors and try to encourage them to order the genomic test. Several of the companies that do sequencing already have committed that they're willing to share that data back with us. And so we also are in the process of creating a data commons primarily for rare cancers where that data and clinical data can be stored, but also will be tied to any tissues, any models that are out there to give kind of a fuller representation for patients and for research. Something that will be starting at the end of next month is custodial biobanking. So if a patient wants to store some of their own tissue, Again, particularly in rare cancers, sometimes it's challenging to get tissue out of a hospital, even though a patient may need it for clinical care at another institution. So there will be a charge where the patients will cover our cost on that, but we will have custodial biobanking. On the right-hand side are things that our work is helping to create is models through working with various research partners, the data sets. We do have part of the organization that is also helping precision medicine for particular patients, and there'll be more information about that forthcoming in the next few months. It's been a program that we've been working on for 2 years. Quite frankly, the initial patients that did that, where we basically helped get them personalized vaccines and some other kind of one-of-a-kind therapies, was for fairly wealthy people that could afford it. We now have found a way that we can bring the price down significantly to help those patients. We'll also be doing that. And again, all of this hopefully helps make pathways to cures. And then the learnings and data that we collect will be in open source data commons. So some of the things, whether you work through us or you work with an academic institution or you work with another group in terms of getting patient tissues, there are a variety of considerations that need to be made. Key are regulatory ones. And anything that's doing with human research studies must comply with the regulations and ensure the rights and welfare of participants. So all of these studies require an institutional review board review and approval. And in most cases, that means they need to review the protocol. There needs to be an informed consent. Recruiting materials that are used to recruit patients have to be approved, and security measures to protect any of the patient identifiable information needs to be reviewed. And you can utilize, like most hospitals, academic institutions have their own IRBs. And when we created Pattern back in late 2016, we used Dana-Farber because we were shipping samples to the Broad Institute in Cambridge. And Dana-Farber is an excellent IRB, but we couldn't change one word on a social media post without going back through them, like literally one word. If we had changed a word and not gotten their approval, technically it was a violation. So we worked with the Dana-Farber IRB for the first maybe 2 years. And then as we started expanding our reach and working with a lot of other research partners, it just got too cumbersome to try to keep track of every different IRB. So we, for our work, have switched to an independent IRB. There are a number of them. We are currently working with WCG IRB. And so we have a protocol and a consent that allows us to ship the samples to any of our research partners. We just sign them up initially with the IRB under our protocol, and then it just streamlines the process. Whenever we want to make changes to materials, whenever we want to change the website pattern.org, we're able to do that in a streamlined way. And we're also getting much smarter about the language we use, so that now every time we add a partner, we don't have to go back through the IRB to get them to approve that. Every time we change a word, we don't need for them to review that. Also, if you weren't aware, there are different approval processes for the US and Canada, like WCG IRB. You can get a US protocol and you can get a Canadian protocol, but they're two separate groups and two separate costs for that. And then if there are any protocol violations or incidents, those have to be reported back to the IRB. You have to follow federal, state, and international policies and guidance. Currently, we're not working internationally, so federal and state, but trust me, those can change. And so you need to work with an IRB that is familiar with these things. Like one area, particularly if you have pediatric patients, is the age of consent is different in different states. So you have to make sure that you're covered in the state where that person is consenting. What we do to try to make that easier is we follow one process. We make sure that anybody under the age of 21 that besides the consent online, we also verbally talk to the guardian. Usually it's a parent. It is critical if you are getting patient tissues that the legal guardian has agreed to it, particularly if the parents may not be married. There have been times, in fact, our PI for the pattern study he's a pediatric oncologist, and if you don't get the legal guardian and the other parent isn't happy with the tissue being donated, it can cause quite a liability. So you need to make sure that there's a legal guardian, and then depending on the ages, if the child's of a certain age, you need their assent. Basically, if they're old enough to understand at least a little bit about what you're doing, they also have to sign an assent. And if research is supported by basically any federal funds, at least currently, that follow the common rule, which is basically rules that were created, I think it was in 2017 or 18, to protect patients, you must have an active SAM registration, and you must have a federal-wide assurance. Whoever you're working with that is collecting the samples, just make sure that they have that if they are getting federal funds. If they're not, then basically any tissues that were collected when they didn't have that, there could be a problem with researchers using them. Electronic signatures have different regulations that need to be followed. There may be tissue and data use agreements that need to be signed. If you're planning on exporting any of the tissues overseas, there may be laws for that. And then, of course, with AI coming on the scene, there may be special regulations for that. In terms of consent language, so the actual document that patients or their parents sign, it's really key to put into those documents cost and benefits. So if there's any cost, we don't charge patients unless they want to store their own tissue in the biobank. So you specify if their cost or if they're getting paid to donate any tissues. But it's also important that if there isn't any cost or participation payments, that's put there in there as well. So just be very clear that in a consent that the patient or their family knows. Also, risks to patients need to be clearly spelled out. Even just collecting blood, there can be bruising, whatever, that needs to be in there. They need to know that there is the potential for a breach of protected personal information. Hopefully that is not going to happen, but we have been advised by the various IRBs that we work with that is very critical. And then genomic testing, if that is being done, and if it's being done in a CLIA lab, will any of those results be returned to the patients? In certain cases, they may not be, and that's fine, but that needs to be spelled out. Also, if you are returning that or a group you're working with is returning that, there could be major implications to that family, as most of you in the room know. And so just be aware of that. Vulnerable groups such as pregnant women, prisoners, or those with diminished intellect. We're at pattern.org, particularly for rare diseases, rare cancers, we are able to collect from those populations, but a number of studies may not want to collect from those populations. And there also may be language barriers if our consent currently is only in English. Our website is only in English. So if it's a Spanish speaking only patient, probably we shouldn't have them consent because we're not sure that they fully understand what they're consenting to. Tissue types, ensure that the consent gives the capability for whoever's collecting that tissue to collect whatever type of samples you need. And also look forward in terms of what you're planning to do with the tissue, not just today, but what may be your future needs. Because if you have a patient consent today, and let's say in 2 years, you find that you still have some of that sample, and there's something else you want to do with it that you didn't think about, you may not be able to do whatever that new work is. So try to think ahead if you can in terms of what might be coming down the pike and using language that is broad will protect you the most, so that you can be specific, but you should also have expansive language for things that you may not know about today. Think about commercialization. And if you're planning to commercialize, which I think a lot of people in this room would be, make sure that that is in the consent. And then who has intellectual property rights? We at RCRF, we aren't involved with intellectual property rights, but that is a point that can be kind of sticky with a lot of groups. Look at, as I mentioned, current and potential future research and assays that you might want to do. And really key, because we learned the hard way with our initial consent and protocol, we didn't have in there that we could recontact the patients. It's really critical, we think, with rare cancer, rare disease patients, that there's the opportunity, if a patient wants to opt into this, that you can contact them in the future for additional studies that may come down the pike, either at your company or another company, and also for contacting them to make sure that their contact information is up to date, and that there is a backup contact. Again, with rare cancer patients, some of them may not be alive in a year or two, and so you need a backup contact if you have some of that tissue remaining, or there's a finding that has come out of the research. If you and your company or your foundation have decided that you want to be able to give that information back to patients, and it's, again, fine if you want to, fine if you don't, but make that decision before the patient's consenting. And as I mentioned, for research-only results, historically, you haven't been able to give those back to patients. There have been some situations where now you can. So make that decision up front if that's something you want to be able to give back to the participant. Logistical considerations for tissue collection, since we collect literally from anywhere in the US, including Alaska and Hawaii. We are technically not a covered entity, but we still follow HIPAA because we feel that's what we need to do to help protect the patients. We assign participant and case identifier codes so that the researcher receiving the tissue doesn't know who the patient is. We are starting to use GUID coding, which is a special participant code. One of the challenges with those are there are various companies that do that. That helps track what studies a participant is participating in, but with different companies using different codes, it's a little tricky. And so we're going through all the learning process of that now. And then some studies, particularly some that have gone with NIH, there was a Human Cancer Model Initiative program that we were a part of a few years ago. Models that were created from samples that we sent to the Broad Institute that are now at ATCC. We aren't able to tell which came from our samples because they mandated that information was delinked. Sample transfer requirements, depending on what type of samples you want, make sure you're clear as to how you want them sent, whether they're frozen, ambient, refrigerated degrees. Make sure you're clear with who's ever sending those. Saturday deliveries are kind of a big thing in our world because there are a lot of surgeries that occur on Fridays, and most labs aren't open on Saturday. So you have to make sure what's going to happen with those Saturday deliveries. Contamination risk reductions. There are some labs that don't want to deal with certain diseases because of potential contamination. So just be aware of that. Post-mortem collections, we get asked about this a lot. They're doable, but they are very challenging, particularly if we're looking at solid tumors. And home collections, where blood draws are usually done. Really the only risk that we have there is if whoever is doing that blood draw does not cover up the label as we instruct and leave if the kit went directly to that patient's home and their name and address is on there, that's not good if it gets to the research lab because that's technically a violation. So you just have to make sure you cover those bases to make sure you try to reduce the chance of that. And then if required, sign material transfer agreements and use agreements. The way we get tissue to various research projects is through the use of a fairly basic material transfer agreement. And that's it on Pattern. We really, just like Irene said, we put patients at the center of everything we do. And we appreciate your time listening today.
Thank you, Barbara. So we having just heard about the power of empowering patients to help assist us in the discovery process. Our third speaker today, Dr. Emil Pindon, is going to share some insights into the complexity in models and preclinical platforms in rare disease. Dr. Emil Pindon is the Chief Scientific Officer of the Myhre Syndrome Foundation. She earned a PhD in neuroscience from Paris-Orsay University and has conducted research at renowned hospitals in both Paris and London, as well as having a extensive career in the pharmaceutical industry. As project and group leader in preclinical drug discovery, Emil has honed skills in cloning, molecular pharmacology, and innovative screening technologies. In the past decade, she has dedicated herself to rare disease, supporting patient-centric organizations with drug discovery strategies. Her work includes creating preclinical platforms and tools to help develop tailored treatments and options for patient needs. So welcome, Emil. Thank you.
Thank you. Thank you very much for having me at the iXCells. So I'm going to focus more today about multisystemic rare disease. That's I think something that is quite common to different diseases. Thank you. No, that's not the right one. Okay, so Myhre Syndrome Foundation is a patient advocacy and research organization that started in 2019 with 2 patients getting together. We are focused on the 2 aspects, community service, but also research. And recently, the research aspect has been really focused on drug discovery. So we want to have the right path and the right tools and how to choose the relevant drug discovery tools. Whatever the goal is to improve quality of life or decrease the life-threatening events or cure the disease, what we need is really to identify targets, models, and assays. So starting with the models, what everybody wants to say is, okay, we want to screen drugs and validate drugs in a model that recapitulates the disease. Well, that's not for us. Because unfortunately, we have multisystemic disease with a lot of heterogeneity. And this is a common issue with rare disease genetic disorders. So the molecular origin of Myhre syndrome is a mutation in the SMAD4 gene, which is also called co-SMAD. SMAD4 is a transcription factor integrating the TGF-beta pathways, and TGF-beta is a superfamily with 33 members and 12 types of receptors. So that's at the end of a lot of different pathways. Specifically, Myhre syndrome is missense mutation on only 2 localizations, the 500 and the 496. And the 90% of our patient is comprised in these 2 mutations, and the rest of the 10% is still at the site of the 500 with 3 others mutations. So the particularity of the mutation SMAD4 for Myhre syndrome is that it's a gain of function through an overstabilization of SMAD4 and the other SMAD complexes. So a gain of function of any kind of TGF-beta will act on a lot of different cellular and functional particularities. So from cell differentiation, and from people who do stem cells differentiation, you know that there is the SMAD dual inhibitor protocol. cell growth, cell proliferation, extracellular matrix production, angiogenesis, tissue repair, immune regulation, and apoptosis. So that's a lot. If you increase the function of all of that, that's a lot. In our case, we have to be very careful in the treatment we choose and the methodology we choose, because SMAD4 has an haploinsufficiency, and the loss of function is responsible for other diseases. So we need to decrease this over this gain of function and over stabilization, but we need to be careful not to lose too much of the SMAD4 activity. So just to show you the complexity of the situation, the superfamily of ligands is TGF-beta, bone morphogenic proteins, BMPs, the activins and inhibins, nodal, anti-Müllerian hormone, growth differentiation factors. So that's a lot. And you have different type of receptors, which are classified in two types. The type 1 is the ALK, so the activin-like kinase receptors. And the second group is the type 2, which is the BMP, anti-Müllerian hormones, or TGF-beta receptors. So you have a dimerization and then an association between the two type of receptors. And then you have the signal transduction that we call the canonical signal transduction from TGF-beta that goes to the SMADs. So some of them will go to the SMAD2 and 3, and other will go through the SMAD1, 5, or 8. But at the end, all of them needs the SMAD4 to form a complex, enter the nucleus, and act as transcription factors. The thing is that this complex with SMAD is only a half-unit transcription factor, and you need the other half that can. We have now over 40 cofactors identified with the SMAD complex that goes from beta-catenin, TCF-LEF, with signal. So many different other signal transduction associated that we need to form the second half unit of the transcription. So you can imagine that we have a huge cell type and context dependent activity of the SMAD4. So from there, you can imagine that we have a multisystemic disease. We have neurodevelopmental deficits with intellectual disability, autism, speech delay, and nonverbal patients, some nonverbal patients, skeletal abnormalities, neuromuscular, also, it's not only skeletal, it's it's musculoskeletal, sorry. We have fibrosis, whoops, that was the wrong one. We have fibrosis and stenosis that trigger life-threatening, and in fact, the last 6 months, we had 3 deaths in kids because of tracheal stenosis and aortic stenosis. And we have widespread fibrosis in various organs, even in liver fibrosis. We have hearing loss, cardiovascular condition, respiratory condition, gastrointestinal condition, immune dysregulation, endocrine, reproductive system abnormalities, and even solid tumors. So from there, you say, well, what system am I going to take, and on what model should I focus on? But that's not all. If you find a system and you say, okay, I'm going to that system, we have heterogeneity in the system. So we have, okay, we go for neuro, we have almost 80% of seizures, but that's cumulative. You can have several of those conditions, but not all of them. Every patient is going to be different. The developmental delay seems to be something quite common. So if you go to other type of system, for skeletal or hearing, the same, you are going to see, again, a lot of heterogeneity of the phenotype that you can have. And in hearing loss, even if you have 53% of the patients that have hearing loss, we have half of them that are conductive, but we can even have some rare cases of vestibular schwannoma, like in NF2. So that's really a big issue on the model selection. Same for cardiovascular, respiratory. I'm not going to show you go through all of them, but a lot of heterogeneity. So what can you do? Where do we go? Well, we decided to go to the patient and look at the patient priorities. So through surveys, we identified that the cardiac manifestation and respiratory were the main two issues where the patients wanted to have some drugs. So I put in 4 fibrosis and stenosis because in fact the cardiac manifestation and respiratory are mainly due to fibrosis and stenosis. And the third aspect that the patient really wanted to focus on was behavioral. And if you go to the one of the patients that was mentioning what altered in adolescence was a shift in his cognition, and he developed psychosis. This is difficult to manage as a family, much more challenging than heart surgery, which he's had. So the behavior was really a big issue, even bigger than some cardiovascular. So we decided that that would be our focus: fibrosis, stenosis, and behavioral. To have the model, so we wanted targetability and modelization. The model has to have a continuity and a correlation at every level, from biochemical, cellular, organ, system, or whole organism. So the 3 questions that we ask ourselves is what is the pathway in those fibrosis and behavioral, the mechanism responsible for the phenotypes we have. Is the model sensitive to this pathway and relevant to the application? And do we have reversibility in the model we choose of the phenotype of the disease? Is it possible to have reversibility? Because we have also adults patients. So we applied some model selection criteria, which was we wanted a limited engineering intervention. So we went for the patient-derived iPSCs. We wanted feasibility and timeline, not a model that is going to take 1.5 years to get ready. Reproducibility and standardization for the screening. A good throughput, so that it won't take 6 months to test 100 compounds. We wanted an assay available or easy to further develop or validate in our models. And we wanted a relevance to the identified targets. So behavioral and fibrosis, that's still very large. Could be, so we ask ourselves, is there a coherent link between behavior and fibrosis? And in fact, we started from fibrosis. And what's fibrosis? It's wound healing that goes wrong, that goes over what it should be. So we thought, okay, that's peripheral system response to injury, that's fibrosis. What's the CNS response to injury and stress? And what's the link with the TGF-beta? So for the peripheral system, if we go for fibrosis, you have the choice of different cellular model. You have fibroblast, of course, that's the first thing you think about. But you have many other cell types, such as endothelial cells, epithelial cells, that all go through reactivity and transformation that we call the epithelial mesenchymal transformation. And this transformation leads to the creation of myofibroblasts, which is the issue in fibrosis. What about the CNS? So what's the response to injury? Well, that's not neurons. That's microglia that is modulating and responding to injury and or diseases. And the other one is astrocytes that also goes through reactivity and scar formation. So we were thinking, okay, so for the CNS, let's focus on CNS. it seems that microglia and astrocytes for our behavioral model might be more appropriate than neurons. So we checked with the TGF-beta and CNS, and we see that TGF-beta is a regulator of microglial function, inhibits phagocytosis and myelin clearance. And we thought, oh, okay, myelin clearance. In our patient, we have in the MRIs, we have some white matter hypersensitivity, which can be a sign of demyelination and myelin issue. So that was one more point for microglia. TGF-beta is also involved in the astrocytes reactivity and synaptic plasticity. It's involved in different pathogenesis of neurodegenerative disease, but that's not our goal here. We have also a pre- and postnatal macroglial cells that is regulated by SMAD4. SMAD4 is critical for driving the transcriptional signature of the microglial cells. So that was, we thought we were going in the right direction, but something that was really important for us was, what about the adults? What about reversibility of the model? Can we have a model that can show reversibility of the phenotype we have? And there is a publication on molecular diversity of the diencephalic astrocytes reveals that adult astrogenesis is regulated by SMAD4. So that was one more point to go in that direction, but we were asking, in fact, okay, diencephalic astrocytes, are we really, do we really want us to have that? Is it relevant for our disease? And in fact, if you look at the diencephalon, which is really at the center here, you see that you have the hypothalamus, which is involved in endocrine system. And those hormones from the hypothalamus are the hormones that are involved in Myhre syndrome disease. The thalamus, where you have motor function that we have issue with in Myhre syndrome, and sleep and sensory perception. So sleep is a huge issue with Myhre syndrome patients. Again, the epithalamus is involved in the regulation of sleep, which is one of the big issue. And we have a certain amount of our patients that are already treated with melatonin, and so some improvement in the sleep. So, and the last part is the subthalamus that is involved in movement, and there are movement issue also in Myhre syndrome. So that was really to reinforce us in the astrocytes. We said, okay, astrocytes too. What about the fibrosis? So fibrosis, I told you, there's a lot of different origin of the myofibroblast. The first thing you think about is the fibroblast. Okay, let's go for fibroblast. And then you see, again, we go from a multisystemic heterogenic disease to a huge heterogeneity between fibroblast, intra and extra organ heterogeneity, and a big difference between fibroblast from human to mouse. So we said, well, could we avoid fibroblast? So we went back and looked at the different cells in the lineage of the cells, and mainly we are affected by all the different MSCs, differentiated cells. So we could go for all those different cells that would, in fact, at the end, give some fibrosis. So we went back again, asking, what about the patients? What's the burden, and what's the big issue for the patient in fibrosis? And the main one is vascular fibrosis that trigger stenosis. So we decided to go for vascular endothelial cells, and the assay will be the endothelial mesenchymal transition and wound healing assay. So we finally had our preclinical platform designed. So we sent the blood immediately to iXCells and got the iPSCs. The in vitro parts of the platform comprise the iPSCs, reprogram vascular endothelial cells, reprogram microglial cells, and organoids. And we are going to work with Alison Morty, hopefully. So again, this name coming up for brain organoids. We have also other organoids for adapted more for fibrosis. The in vitro cellular assay we have binding assays and more biochemical assays with measuring the stabilization or destabilization of SMAD4 SMAD complex here. And then we have the functional assays from the cells, which is, as I said, fibrosis, wound healing, and glial reactivity. We hope that the astrocytes will come when we have more funding. The in vivo models are conditional knock-in mouse model. From one is already available at Jackson Lab, and the other one will be on the way. The in vivo assay will be on fibrosis and stenosis, but we are starting deep phenotyping with psychogenics to really get the right screening assay specific to Myhre syndrome markers in behavioral and other characteristics. So this preclinical platform is really at the center of our pipeline, and it's really fundamental for us. So we have our treatment portfolio for now is composed of small molecule repurposing and new chemical entities, around a bit more than 100 that came from the IRB in Barcelona. Sorry, in Barcelona, the IRB Institute. They were selected, the hits were selected on the destabilization between SMAD4 and SMAD complexes. We have peptides from a company in South Korea. Our big effort is in immunocell therapy, so FAP CAR-T therapy to reduce the fibrosis. And the gene therapy editing had the first good results and is going into the iPSCs now. And the more translational research is on the neoprotein-protein interaction between the mutated SMAD4 and new cofactors, and the new brain organoid with Alison Morty and new therapy in development. And hopefully going through the preclinical platform will allow us to jump into our clinical platform that we are working on. So this preclinical platform, we hope, could be expanded to other TGF-beta related diseases, just that have fibrosis, vascular condition, neurodegeneration, or glioblastoma, or others, where you have, in fact, with Myhre syndrome cells, a kind of natural overexpression. It's not overexpression, but gain of function. So on overstabilization and over signal of all the TGF-beta. So that's, I think, a good way to screen and read any treatment that could decrease that. So that's, so I just talk about the first efforts. That's our preclinical, but we are working also on clinical readiness, and those are the treatments, our pipeline of treatment that we are working on now. And we work also, of course, on the regulatory and industry. Okay, thank you.
Thank you, Amel. Wow. We talk about the complexity of getting the cells, and then when we get them, the amount of work to come up with a relevant model there, it just really hit me. So thank you for that. As we move into the final speaker in this session, it is my pleasure to introduce Dr. Kitchener-Wilson. Kitch is the CEO and co-founder of Rosebud Biosciences. He is a physician scientist with 16 years of experience merging human iPSCs with genomic and molecular technologies, particularly in the cardiovascular applications. As a Stanford-trained molecular pathologist. Kitch was an attending physician at Stanford for half a decade before founding Rosebud. He holds a BS in mechanical engineering, a PhD in bioengineering from Stanford, and an MD from Duke. It's my pleasure to introduce Kitch and his presentation on scaling stem cell-derived organoids for disease modeling and drug discovery.
Great. Can you all hear me? All right. So I am a co-founder CEO of Rosebud Biosciences. We work with iPS cell-derived organoids. This comes from many, many years that I did in academia. I jumped ship to the detriment of my colleagues there to start a company around this. And I do truly think that this has a place in drug discovery. It's at early days for organoids. We've seen a few people mention them. What we really focus on is scaling them and homogenization, precision, quality control in terms of organoids. And it's organoids broadly, and I'll show you what I mean by that. So I show this slide to all the investors that I pitch to. I think this is really what we do. And if you take anything away from my talk, this is it. We have organoids that are mimicking fetal development. Remember that these are iPS cells. Anything derived from an iPS cell is fetal-like. These are not adult tissues. This is not a 60-year-old person's cells in a dish. However, with genetic diseases, you can frequently find a phenotype in these organoids that mimic what happens either in utero in terms of development or shortly after birth. So we introduce mutations into iPS cells. We have our own secret sauce for deriving them in a dish. I'll show you a little bit about that. And then we do longitudinal monitoring every single day. I would love to do it real-time monitoring someday, but we have longitudinal monitoring every day looking for a disease phenotype. This is what they look like in a dish. So this is a 14-day protocol for deriving cardiac organoids. I'll show you a little bit more of that later. These are just bright field images once a day of a developing cardiac organoid. There's this, as far as organoids, I think that there's a lot of, people say organoids. It can mean a lot of things. It could be cancer organoids, maybe little microbiopsies of tissues. For us, these are actually attached on matrigel, so they're relatively flat. So they're not necessarily 3D, they're 2.5D, if you will. So there's a structure to them. However, the reason we like these, and this was a 2014 publication that really kind of set the field ablaze, these are attached circular micropatterns of iPS cells, 2 millimeters, 3 millimeters across, all on standard tissue culture plates. They go through embryogenesis. So you can actually see concentric circles. Something laser. No, that's laser. Anyway, that's which one? Oh, that little one. Okay. You can see right here. So you can get endoderm, ectoderm, mesoderm, all in concentric circles here, and these are regular. So that means the thing is not just a homogeneous group of cells, but they're actually going through embryogenesis. So we like this because you can scale these much more easily than the spheroids that everyone thinks about with organoids. The other thing, before I move on to this, the other thing about this, because they're relatively flat, makes imaging much easier. They are static. They're not floating up around. They're not spheroids, so you're not getting necrotic cores with them. Diffusion of nutrients, drugs are through the entire thickness of these 2.5D organoids. They make them a lot easier to work with. So these are some cardiac organoids. I come from a cardiac background, so you will see a little bit more of a cardiac bent to this talk, but we have more tissues that we do. Single cell RNA-seq. You get, because of that, it's going through embryogenesis, all the multiple germ layers. You're getting cardiomyocytes, you're getting fibroblasts, you're getting endothelial cells. There's even some neural progenitors, which are that rudimentary conduction system with them. So they are very diverse cell type, tissue types. So I think of these as tissues, not as homogeneous cell cultures. There's a little bit of a busy slide. The take-home point for this is rodents, 2D cell cultures have been the mainstay for decades, for probably half a century for drug discovery. You go from 2D to rodents. You do your preliminary tests in 2D, then you go to rodents. What I think is that these organoids are going to bring any complexity, and in the near future, they're going to be that gap between the 2D and rodents. In the future, I really do, I'm a maximalist on these things. I think that they are going to replace all of those, and organoids in the 21st century are going to be the primary drug discovery platform. That may not happen in the next 2 years, but I think in the next 10, 15 years, you're going to start to see these being the mainstay for drug discovery. These are rich, heterogeneous, macroscopic as opposed to microscopic tissues in a dish that have mutations of the patient. They show disease beautifully. I think that as long as we can standardize them, agree on particular quality controls as we do with our 2D cultures, that they really are going to be the mainstay. So this is a little cool video. They lit up our laboratory with some fluorescent lighting to make it look like a nightclub. We're very excited about this. This is what we have. We have a robotic arm. We grow our organoids in 96-well plates. And you can see our robotic arm. We have a microscope in the background that's moving them around each day. So there's a liquid handler. They're getting fresh media. They're getting bright field images every single day. Once we put a plate of micro-patterned iPS cells in these plates into this system, it's hands off. Humans are the biggest source of error in terms of culturing these iPS cell-derived organoids. So we've tried our best to remove the human component from actually producing these organoids. So this is automated. It works on weekends, it works on vacations. So it's wonderful. We're a team of 3. That's the entire team there. We can produce these in the thousands. We have a capacity for about 10,000 organoids that we can do simultaneously. And that includes longitudinal imaging every single day, media changes, you name it. So the real idea is to use this as a drug discovery platform. And of course, my co-founder, who is big in AI, we are training AI. What we want to do is train it on bright field images of organoids, this is similar to what Recursion is now doing, and get away from the need for immunostaining, biomarker staining, that sort of thing. So just training on bright fields. That's our long-term aspiration. We're not quite there yet, but I think that's where we're going to go with it. I'm going to give you 3 examples. So this is dilated cardiomyopathy, which is my background from my academic days. Hearts get enlarged. They're not able to pump blood. There's probably 50 genes or more involved in dilated cardiomyopathy. It can also be environmental, all sorts of issues. These folks generally need transplants within a year or 2 of getting diagnosed. Mortality rates are atrocious, about 50% die. There are no targeted therapies for this disease whatsoever. So this goes back into my academic days. I found a gene that's associated with DCM. It was upregulated in DCM, dilated cardiomyopathy. This was now 10 years ago. And this led to the whole saga of trying to figure out what it does. The challenge with this particular gene is called long banker, which is a long non-coding RNA. It's primate specific, so it's not found in mouse. You can't do a mouse model with this. It's also fetal specific. It's only expressed during fetal heart development. So how do we get around actually studying what it did, since it seemed to be associated with DCM? Well, HESC, this is human embryonic stem cells, iPS cells, are the same thing. Derived cardiomyocytes also express that because remember that they're fetal-like. So I thought, after a lot of work, it looked like migration. It caused cardiomyocytes to migrate based on a lot of RNA-seq data and some other reports that we had seen. So I did a swooned scratch assay, which is usually you do with endothelial cells and fibroblasts. Very unusual to do that with cardiomyocytes because you don't think of them migrating that much. And I found a little bit of a difference. It wasn't very satisfying, however. You can see just a little bit. There's a p-value. I think it was a 0.01. That was probably on a good day. I didn't really think that that was the true phenotype, and I needed something better. So what we did is we moved on to organoids. I had a colleague that was working on these micropattern organoids at the time. And this is where you saw this beautiful phenotype in a dish that you could see with your eye. And this is actually the source of the name of the company because they look like flowers to me. So what's happening here is the green are the cardiomyocytes. All derived from, in this case, HESCs. They've been micropatterned in these arrays. This is using an older technology, stencils, but this is something we've come around with later to make it more high throughput. And we're overexpressing this particular gene. It causes migration of those cardiomyocytes, which you can see kind of like a blossoming flower. And here's our control with the scramble control. This, to me, is what sold the deal for organoids and their ability to show a phenotype far superior to what you would see with single 2D cell cultures. And this is what tells me that organoids have that macro ability to show disease, which you could almost see with an eye, with a beautiful recapitulation of what a disease of in vivo disease. So we've scaled this up. Let's see if this works. Do you see that? So we work in 96 well plates. These are cardioids, differentiating them. Homogeneity is a big deal for us. I'll show you a little bit more about what that means. We've introduced mutations for DCM. So on the left, you can see are our beating disease ones. On the right, I'm sorry, let me see if I can show that again. On our right is the disease lines. On their left lines are wild type. You can see they're kind of squirrely looking. So we're really trying to get high throughput homogeneity between batch to batch with these things. High N is really what we're going for here. This is we'll look at the single cell level, single organoid level. You can get beating rates, you can get magnitude of beating, you can get percent rating, and then you can see the disease at the bottom is the disease isn't beating at all. So beautiful data that you can really get with these organoids. We moved on. You know, starting the company, I did not want it to be just a cardiac company. We're like, there's protocols for liver, there's protocols for kidney, there's protocols for brain as well. This is a brain, I'm sorry, a liver. This is for a disease called Alagille syndrome. You get a paucity of bile ducts in these kids. 60% of these kids ultimately need a liver transplant. There is no targeted therapy for these whatsoever. It's a well-defined genetic disease. However, JAG1 is the actual mutated gene here. So here's a hepatic organoids. I'll just point out, you know, they have vimentin is our fibroblasts. Here is our bile ducts. CD31 is our endothelial cells. Albumin are hepatocytes. So there's a diversity of cell types in these. You can also see secretion of albumin by these. So this is actually the media of these organoids that you can sample and run ELISAs on those. They also metabolize urea, and you can detect urea in the media of these. So they really are functional little units of microtissues. So here is where we introduced a JAG1 mutation that causes Alagille syndrome. And I told you that there's the bile ducts. There's a paucity of bile ducts, meaning these kids aren't able to produce bile ducts and move bile through their livers. And so here is wild type, where you see actually normal ring-like bile duct epithelium. These are a few millimeters across, but you can see that regularly. This is a stain for actual bile ducts. So that's wild type. Down here, you see our mutated line. Virtually gone. There might be a few little hints of bile duct here, but for the most part, they're gone. This is something you would not see at the single hepatocyte level or the single bile duct level. This is something you see at the macrostructural level. And so this is where organoids really come into play. Third and final example, Fabry disease. So this is a disease, there's actually a lot of interest in this disease in pharma. There's quite a few companies working on this. There's shortened lifespans for these folks, about 50 years for men and 70 years for women untreated. What it is there's a it's a lysosomal storage disease, multisystemic. You have a healthy GLA gene. It produces alpha-gal enzyme. It metabolizes this sphingolipid called GB3. When it's mutated as GLA gene, you have low amounts of this alpha-gal gene, and you get buildup of GB3. This could be multisystemic. Typically, what these folks die of is either heart failure or kidney failure, but it's multisystemic, so it can occur in lungs and elsewhere. A lot of interest in this. So we were approached by a pharma company that wanted to model this. Apparently, the mouse models do not recapitulate the disease pathology to their satisfaction. And so we said, okay, we have human organoids. Let's try that. We knew nothing about kidney at the time. We developed a protocol for developing kidney organoids in a dish. We introduced 3 different clinical mutations in the GLA gene that are known to cause Fabry disease. We also have a knockout line. GB3 is our marker for the buildup of that sphingolipid, which causes disease in Fabry disease. And then there's a variety of kidney markers. You can actually get little proximal tubules in here and podocytes too, which is pretty cool. So this is what they look like. On the right is what a healthy kidney organoid with a functioning GLA gene that metabolizes its GB3. This is a stain for GB3. On the left here, see all these red speckles? There's a bit of red background here, but you can see those bright red speckles. That's that buildup of those sphingolipids. And so we could get this in this was about a 3 to 4 week protocol from start to finish, and we could produce these in the thousands. So that's been exciting for us. We can now do drug screening and all kinds of things with this. These are human, remember, these are not mouse models. So this is just an overview of what we are focused on as a company. I've shown you heart, I've shown you kidney that we've done. We have this liver program. We have an SBIR funding that, although we'll see if that continues these days. And we do have some interest in Huntington's disease and brain. We do have some preliminary data for brain organoids too, although it's kind of early days for that. Ultimately, I want to do all of these tissues. If there's a protocol for differentiation, you can do modify it for a protocol for organoids. We are not spiking in cells. We're not creating 3 different cell types and mixing them together to create these. These are all assembling by themselves as iPS use in our 96 well plates using our different factors that we add to the media. So relatively simple. If you ask AI, what does this is a Grok actually, what does an automated organoid factory look like? And it gives you this. It looks a little dystopian, I'll be perfectly honest. And I think my father had one of these in the 1970s for playing music. But there's some things about this that I do like that captures where we're going for Rosebud, our company. As a pathologist, you think about precision. And precision is consistent between repeated measurements. And that's always been an issue with biology, and particularly when you're having humans culture these cells. If you remove humans from, I love humans, but if you remove them from the actual work of producing these organoids, you get better batch to batch and repeated measurements. Accuracy, are they close to the true value? There's a very low signal to noise in a lot of biological research. We're trying to increase that delta between signal and noise. And so this automation helps that. High throughput, of course, pharma wants high throughput. They don't want little 6 well plates of single organoids. They want thousands, preferably tens of thousands, to really produce high-end statistical value. So that's what we're going through here. Modular. Technology changes. Microscopes get better, liquid handlers get better, robotic arms get better. I don't want a $4 million box that I have to buy from a company that has everything that breaks down. By the way, I have to send a technician to come fix it, but that is stuck with the same microscope, stuck with the same liquid handler, and I'm never able to upgrade. So I like the modularity that this actually image shows, and that's what we're going for as well as company. Interrogatable, meaning you can change the conditions, change the media, change the factors, change the environment of these organoids to produce different data to create disease organoids. And then real-time monitoring. I really think that it's important to detect disease as these things are developing in a dish. And when is the moment when you can first see that detection of disease and that diverges from health? And a lot of AI is going to be involved in this real-time monitoring. So I think that's where we're going. I'll wrap it up here. I don't think this audience needs any introduction to rare diseases. I tend to use genetic disease for iPS disease modeling. There needs to be a genetic component, in my opinion. If there's no known genetic mutation or gene involved in the disease process, I would be hesitant to get into it because it could be environmental, and it's tough to decide what is that environmental perturbation going to be to elicit the phenotype that you want. So I call it genetic diseases. And rare diseases, I think it's about 80% are known to be genetic. So that's my preference, to call them genetic diseases. Our goal, high throughput, precise, accurate phenotyping of the human genome for target drug discovery. And our working hypothesis for this company is that 3D tissue is greater than 2D homogeneous cell types for disease modeling. And human data is better or equal to rodent models. I think ultimately they will be better, and that's our competition are rodent models, to be quite honest. What these do lack, however, is immune system. There is no immune system in these organoids, and that's true of any 2D culture. So we have to find creative ways to get around that in terms of modeling the immune system. So we're sticking with genetic diseases of actual organs for now. This is my team, myself, Evan, and Aditya. We are a Y Combinator company from 2 years ago. We have a few investors in it. We're based in San Francisco. If any of you are interested in organoids, please reach out. Thank you.
All right, thank you, Kitch. We have a few minutes for questions. We'll take questions in the room first. If I could just ask the speakers just take a seat at the front, just makes it a lot easier with questions. Okay. Wow. Okay. Just take that one over. I'll pass this microphone over to the panel. Thank you.
Thanks. That's great talks, everyone. I've got one really sciencey question for Kitch, and then a broader question for this group. Think about standardization. Kitch, it was great seeing the single cell RNA-seq data with the cardiac tissues, especially. Thinking about that and the interplay between different cell types driving disease, have you guys considered omics-based or CRISPR pooled screens to look at what individual subtypes are driving phenotype? I was especially thinking about your cardiomyocyte single cell type not showing phenotype, but when you mix 9 cell types, you see a very striking phenotype.
You're thinking about Perturb-seq and
Perturb-seq.
Yeah, we'd love to do it. It's big money for a small startup, but yes, we'd love to do it. High throughput CRISPR knockouts of 25,000 genes, seeing what each one does in the different subcellular types. My ultimate goal, like a pathologist, was to have single cell RNA-seq as our disease readout for a lot of these organoids. It just comes down to cost right now.
There are some newer ways you could think about the inducible Cas system in your IPS that you then turn on once you're in your organoid stage.
Absolutely.
Throw your pool in, do a crop seq and look at.
We'd love to do that. Yeah, absolutely.
And then broader, everyone mentioned standardization, scalability, reproducibility. Leveraging AI and thinking about all these machine learning techniques, we need to have some sort of commonality to start pooling data together from multiple different platforms. What do you think about the scientific industry needing to do differently so that we have shareable, combinable data that we can start to identify patterns? And rare is no longer rare. Rare is just a different way to get to 5, 6, 8, 10 phenotypes.
I think you put the finger on the words sharing data. I think that will be the main issue. That is already a big issue to share data inside a single community of rare disease. So I have difficulty imagining how we are going to do it in between different community. Maybe there will be less competition between different rare disease, so the will to share will be bigger. But that's it's not only from the scientists that we have issue sharing data. It's more on the side of the IP and all the legal aspect and the legal team in academia, university, or I think it's also an aspect in CRO about sharing data. Then you decrease your IP. So that's I think the next challenge, and I have no idea how to solve it.
I can add a little bit because at the Children's Tumor Foundation, we actually have a data portal hosted by Synapse. And so over the last, I guess, 10 years or so, we've been first encouraging researchers to share their data, funded data, back into one central repository, but more recently we're demanding it. So in recent years, it's part of our funding kind of award letter where researchers need to share their data back, and we have it in a portal. But to take it one step further is, now we need to share it outside of CTF, and so we have initiatives where we're identifying complete data sets that are shareable and putting them on public platforms like CBioPortal is one of them we're working on, maybe Geo at some point. So we first have to identify which data sets are well annotated and formatted and go through all of that work, and then identify which public platforms to share them on. And we're actively doing that now. So
I'll just second that, making it mandatory. Like, I've worked with a number of research groups that said they didn't want to share the data. And I said, that's great, but then you're not going to get tissues through us. So that changed their mind, and every single one of them said, okay, we'll work with you. I think also patients are key here. Patients understanding the value of their data, patients getting the data if you can't get it, directly from the researcher, and patients starting to talk to people about, I want my data shared, I think is really key.
Yeah, second all that. In academics, you have to, there's Geo and all the repositories for seq data, but industry side, it's a mess to keep all this data proprietary. And then our business model is partnering with pharma, who themselves don't want to share that data because also the iPSCells themselves, there's incredible royalties and fee structures and proprietary. You can't even screen for molecules without paying Japanese academia for the IPS academia for these things. It's onerous, the amount of boundaries that they place around iPSCells. So what we've got a workaround solution. We're able to create our own disease lines and then generate data from those. I would love to put that data somewhere in the cloud that's accessible to all. Right now, it's accessible to only our partners, which is unfortunate. But yeah, there does need to be some sort of industry-wide paradigm for addressing that.
Okay, I have a question, Kitch. So you talked a little bit about this, your 2.5 versus 3D organoids. But did you see any differences? Have you ever done experiments that shows really the outcome or phenotypes? Is what you see only possible with the 2D versus the 3D?
2.5D versus 3D. I can only tell the practical differences we've seen. They're relatively flat and they're attached. The 3D, they are floating in media. They are not able to get oxygen and nutrients into that central core for those spheroid type things. And so many of them get hollowed out, which can be a challenge with those. And then imaging, meaning that, you're taking serial imaging of these things, because those spheroids are floating in media. Each day that you're taking an image of them, it could be in this orientation, it could be in that orientation. So comparing it to between days is also a challenge.
Yeah, no, I get it, but there are some people that kind of optimize, standardized to a little bit at least, organoids when they're free floating, right? So did anybody do comparative studies between what you do and what?
I don't know that. I'm not aware of anybody who's done actual comparison of the 2D versus 3D, other than the diversity of cell types seem to be the same in the 2.5D versus the 3D. You have smooth muscle cells, endothelial cells, fibroblasts in relatively the same quantities as what you'd see in 3D. They all beat. I showed you a lot of the beating. So beyond those metrics, are they more or less mature? How do you define maturity? We can let these culture for 2-3 months if we had to. I'm quite confident they would be similar to the spheroids themselves, which can be matured as well. I honestly don't see a lot of difference, but if there's been a statistical quantitative study of the difference between the two, I'm not aware of one yet.
So I have a Okay, I wanted to ask 2 questions. I guess I'm going to ask one only. Okay, I'll ask the second one later then. I have a question for the NF1 and the Armile, no, that you show that there is a lot of heterogeneity on both those diseases, no? In the case of the NF1, you show that there is so much phenotypic, no? I mean, in the symptoms of the people and also for the SMAD one, for the TGF-beta. So do you think that's going to make more difficult to find like a common therapy Or do you think that we're going to need to find different therapies, I mean, kind of subcategorize inside of the disease, no, different symptoms? And then do you think that we are going to need to find different therapies like small molecules or whatever for different these categories? Or do you think that it should be able to find something that encompasses the whole disease?
I think you're absolutely right. It's very heterogeneous with a broad spectrum of manifestations. So it will take multiple therapies and mechanisms. You know, we're just talking about NF1 today, but there's a whole other schwannomatosis type that I didn't have time to get into today, but absolutely, it's going to take more than one kind of therapy and more than one mechanism. Ultimately, combination therapies are something that are being looked at. So I think a combination therapy is on the horizon as well, to be able to hit multiple targets of the disease.
Yeah, I think it depends what you target. For us, if you target the SMAD4 or SMAD complex, then you can imagine that whatever the cofactor is next to it, you are going to act on all the different phenotypes. In our pipeline, we foresee that maybe we won't be able to do that. So we are also developing with the Geneton a FAB-CAR-T, especially for the fibrosis, in case we cannot get to the development of the small molecules. But since we start our first screening on really the destabilization of the complex, then the only limitation would be the delivery of the molecules, if it goes through the BBB or not. But if we would go for other like ALK inhibitors, for example, ALK5 inhibitors, that we also want to test, that would be only a part of the phenotypes. But going really at the origin of the issue, I think will allow us to go larger in the targeting of the phenotype. It depends where you go in the signal transduction. And since SMAD4 is really at the end of everything, then if we go for that, we hope that we will be able to tackle many different aspects.
Okay, thank you. I'm just gonna, one quick question from the online team, and then I'll get you all to lunch. Kitch, there was a question in regards to cost. How does the cost compare for the 2.5D versus typical cell and animal models?
Did you say cast?
Cost.
Oh, cost. And between that and animal models? Oh, that's an easy one. They're far cheaper. They're each organoid is less than $5, excuse me, to produce. And you can produce these, you know, in the thousands. So you can imagine the scale that would be impossible with these sort of things. So yeah, they're quite a bit cheaper.
Wonderful. That's great news, because drives the economics of the screen.
Indeed.
So again, I just want to thank all four presenters for a wonderful session. Thank you all for your attention and questions. Again, round of applause for our presenters.